We are investigating Alzheimer's disease. It's the most common neurodegenerative disorder. We are investigating this disease from a different angle than usual. We are focusing specifically on sex differences because many sex differences have been reported for this disease. Females develop the disease more often than males, so there are several gender differences that are very pronounced. And we think that understanding these differences can not only help us to better understand the disease, but potentially also find new targets for therapeutic development. In humans, we can only investigate the late stage of the disease, so we cannot look into the brain of a living patient, of course. At the same time, it's very important to understand the early stages of the disease. And so for that, we need model systems like mouse models or cell culture models. Mouse models are very good because there's a high similarity to humans in terms of the genome, and we can also analyze behavior in these models. If you want to analyze the modulation of individual genes, then it's better to use a model that is a bit simpler than mouse models. So for example, zebrafish models, we can look at zebrafish larvae that are actually transparent. So we can look at these cells using the microscope and actually see what changes. So all these different model systems have different pros and cons. So we basically need to combine all these different data sets to ensure what we find is relevant to humans, but it also captures the really early stages of the disease. There are different types of pathology in Alzheimer's disease. There is the so-called amyloid beta pathology, and there is the tau pathology. Tau and A-beta, these are two different proteins that both aggregate in the disease, and they both play an important role in the disease pathology. But you need different model systems to study both of them. We used mice of both genders to see, okay, are there some sex-specific differences that can point us, in particular, the early sex-specific changes that occur in the disease. And then, of course, we compared these two different model systems, and we used the data to compare it to human data, allowing us to see, okay, are there some changes that may occur early in the disease that we also see in the late stages in human biosamples, so where we can be sure, okay, they are relevant both for humans, but also we have the evidence that they are important in the very early stages based on the mouse model. And so that basically gave us a selection of candidate genes that we then wanted to further investigate, really using perturbation experiments. So basically modulating the activity of these genes in a dedicated model system. And so for that, really, the zebrafish model is probably the most suitable model. It basically allowed us to look at the small selection of these most promising genes and then see, okay, what happens if we also perturb the activity of these genes. First of all, what was quite interesting is that across the different model systems, we could really see big overlaps. So there were some genes where we really see significant changes. So that really gives us confidence that these genes are relevant also at different stages of the disease, and that these genes are also relevant with regard to different types of pathology that occur in the disease. And the data also allowed us to interpret changes in the context of prior pathway and network knowledge, because these genes do not act in isolation, but they interact with each other in terms of cellular processes and signaling pathways, where basically the information flows through functionally related processes, where we also know that they are altered in the disease, and where we basically can interpret these changes in the context of these coordinated cellular process changes. So that was, for me, the first most important result. We can see significant changes, and we can also link them together in cellular pathways and across different model systems. The next logical step is to further confirm this in a larger set of human biosamples. We already did some comparisons to human samples before, but the sample size was relatively small. Ideally, you want to confirm these changes not just in one type of tissues, but across different tissues and body fluids. Additionally, we also want to look at other types of molecules. There are all sorts of different biomolecules that play an important role in the disease, so proteins, metabolites, and other types of molecules. And to really understand how all the different molecules act together, we also want to look at these other types of biomolecules that are also relevant and see how do they interact together. This project has the potential to really identify key genes, proteins, and other biomolecules that first of all play an important role in the disease, and where we see significant differences associated with pathology and sex differences, where there could be basically modifiable factors in the disease that can be modulated using pharmacological approaches or potentially also nutritional interventions, where there is a potential really to achieve a disease-modifying effect.